progress report to understand the same day?
Probably not.
We should
All right.
We should take
So chapter 11 is all about meiosis. Now that name, meiosis, should sound kind of familiar because it's a little like mitosis, and we've covered mitosis. So before we get into the detailed stuff of this, God bless you, we are going to no, let's go. Before we get into the details of this chapter, we're going to review mitosis because you need to have that in the back of your mind. A lot of the stuff that we saw with mitosis, we're going to see again with meiosis. They're different processes, but they have a lot in common. And so that's what this whole chapter is about. We'll point out what we've already seen with mitosis. We'll point out how meiosis is different, but that's what this is. So we have to talk about how reproduction works, especially with sexually reproducing offspring in organisms. We'll look at what meiosis is about and how it works, and then just get a good idea of, more specifically, how meiosis is different than mitosis. So before we get into meiosis, let's review what mitosis looks like. As part of the cell cycle, right? When we look at the cell cycle, it's divided up into what kind of parts? You have interphase. What's going on in interphase? You have G1, S, and G2. What happens in G1? Growth. What else?
Wait, G1? G1 is growth. Isn't that like where you find out if that's the growth check? Oh, that's where it's like...
There's a checkpoint before we can move into the S. Sure. Cells like living its normal life, doing its job, running metabolism, right? Like functioning, keeping your body alive. Eventually it grows so much that it will probably pass that checkpoint. It'll pay attention to growth factors, which trigger the progression through the cell cycle. And then you enter what? S. Synthesis. And what happens in S?
Each chromosome replicates to produce sister chromatids.
We're copying our DNA, we're replicating DNA. Now, how many chromosomes do you have?
46.
You have 46 chromosomes. And so we would have to copy all the DNA of all those chromosomes. But we're talking about chromosomes, and in reality, your DNA does not exist as chromosomes during interphase, right? What form of your DNA is present during interphase? Chromatin with an N, yeah. That's that uncoiled.
Please report to the office at this time.
I'm on my way.
That's that uncoiled, sort of unorganized DNA that exists. We don't see that during mitosis, though, because that would be difficult to separate into different cells. Anyway, after the S phase, after that DNA gets copied, we move into what? G2, right. And what's happening in G2?
It's preparing to divide.
Cells getting ready for cell division. So some of those organelles will get copied, or some actually just disintegrate and go away. But some of the organelles, like the centrioles, get copied, so we have more than one copy of it. And cell grows a little bit more. It's getting ready to divide. So once we move past interphase, we get into mitosis. And that is made of, well, we when we learned it, we said 5 stages, right? So you have what first?
Prophase.
What did we see going on in prophase?
The spindle forming.
You have spindle forming. What else?
The nuclear envelope was...
The nuclear envelope is disappearing. What else? What happens to the DNA?
Wait, it's chromosome prophase?
Chromatin organizes into chromosomes, so we'll start to see chromosomes forming, yes. Well, I could draw a human cell with 46 of them, right? No, it's too much. After we move through prophase, we go to prometaphase, and that is sort of that is a in-between phase Between prophase and metaphase, that should make sense. But what's happening in prometaphase? Oh my gosh, I can't spell. Yeah, the spindle attaches to those chromosomes, and now those chromosomes are moving because the nucleus is gone now. This is really quick drawings for me because I'm trying to get through this quickly, but well, we're not lined up in the middle just yet, so let's not draw it like that. But the chromosomes are attached to the spindle, and the spindle can be pulling them. Then we move into what? Metaphase.
That's where they're all lined up in the middle.
Now they're lined up in the middle. So my chromosomes will be aligned in the middle of the cell. And they're like lined up perfectly. Remember, if we have 46 chromosomes, we would see 46 of these lined up in the middle of the cell, right? So that's a little harder to look at. But 46 of those individual chromosomes lined up in the middle of the cell. And then what happens in anaphase?
They split.
They split. What splits? Okay, so the citrochromatids split and they're moving to opposite sides of the cell, right? The spindle's tugging on them, moving away from each other. What holds those chromosomes together? Well, at the centromere, what's the name of that protein? You remember? Cohesin. And so when the cohesin breaks down, essentially, those sister chromatids move away from each other. And they're moving to opposite sides of the cell. The cell elongates a little bit as well. Then we move into telophase. And what happens in telophase?
Spindle fibers are gone.
Spindle disappears. Nucleus reforms. What happens to our DNA? Uncoils. It uncoils. Opposite it condenses. Right. But we wind up with one cell so far, but with two nuclei, right? All right. So we don't have two separate cells yet. But that's the end of mitosis. What happens during the last part? What's the last part called? That's cytokinesis and the actual cell split. So the chromosomes have already split, but now we'll wind up with two separate cells with their own nuclei. What can we say about these two cells when we compare them to each other? They're identical. They're genetically the same. They have the same exact copies of DNA in them. They're also genetically the same as this first cell that we started with in interphase, right? So we have taken a cell and made an exact copy of it. That's what mitosis does, right? Takes one cell, copies DNA, makes another cell exactly like it, right? All right, when we look at how the chromosomes move around, we see each chromosome kind of moving by itself, right? Lining up by itself, and then the two halves, the chromatids, getting pulled apart. Yes? Mm-hmm. Okay. Just wanted to make sure we notice that, because when we get into meiosis, we're going to see how that's a little different. And we got to be able to pay attention to the differences. So before I get into what meiosis is, we have to understand the whole point of meiosis. The whole point of meiosis is to make gametes, which are our sex cells. They're sometimes called your germline cells. But that's what we're trying to do here. We're making gametes. Do you remember why your gametes are different than your somatic cells?
The somatic go through mitosis, and then the gametes go through meiosis.
Yes. They need different processes for forming, right? Somatic cells, when you're making a copy of a cell, you're just running mitosis. Meiosis is different. It allows us to make those gametes, but what's different about those cells that requires us to have a different way of division, a different type of cell division?
I don't know. One is diploid, one is haploid.
Uh-huh. One is diploid, one is haploid. Do you remember what those words mean?
Diploid is two sets, and haploid is just one set.
Okay, so our diploid cells, like basically, when I say diploid, I mean it's like every single cell in our whole body except our egg cells. Every cell in our whole body except our egg cells are diploid, have two sets of chromosomes for a total of how many?
42.
Total chromosomes. Two sets, a total of 46 chromosomes. That's what diploid is.
That's a human.
Yeah, we're talking about humans.
So wait, gametes have what?
I didn't get there yet.
Regular?
I'm talking about somatic, yeah. All your somatic cells, which again is every cell in your body except for your egg cells, they're diploid. And that means they have two sets of chromosomes for a total of 46.
Did you
But-
lecture at all last time?
No, I wasn't here.
Okay.
when we look at our egg cells, those are haploid, which means they only have one set of chromosomes. So that would be a total of how many chromosomes?
46.
Nope,
23.
23. Diploid is 46, haploid is 23. So our egg cells have one set of chromosomes, that's a total of 23. Now, why do our egg cells have to have a reduced number of chromosomes? Well, that's what we're contributing to our offspring, right? So we use our egg cells to make offspring. Our egg cell has to combine with a male sperm cell to make offspring. So we have to be able to combine those chromosomes to get the correct number of chromosomes in our offspring. So our egg cell has to have 23 chromosomes, sperm cell has to have 23 chromosomes, so when you combine them and make your offspring, you get a total of 46. That's the way reproduction works for us and for all sexual organisms, right? And when I say sexual reproduction, I don't mean sexual intercourse. I just mean fertilization has to occur. Sperm and the egg have to fuse. So that does mean that you have male and female, and that is handled in different ways because like plants actually reproduce sexually too. But they produce sperm, they produce eggs, and fertilization does occur. But that's part of our life cycle. And so in order to make these gametes that work correctly and they have the correct number of chromosomes, we have to have a way to reduce the number of chromosomes from the diploid 46 chromosomes to the haploid 23 chromosomes, because we have to get this and this to combine to give us that total of 46. If we had gametes that had 46 chromosomes and then they combined, well, we'd wind up making cells that had 92 chromosomes, which obviously would be way too many. And too many chromosomes, just like too few chromosomes, is a big problem. We'll talk a little bit about that later, actually. So if we understand how mitosis works, that gives us enough background to walk through meiosis and understand how that works. But remember, the whole point of meiosis is to make these types of cells, to make these haploid gametes, which means this cell division has to reduce the number of chromosomes. In meiosis, we are not trying to make exact copies of cells, because the way meiosis works is we start with a diploid cell. We start with a cell that has two sets of chromosomes. And then we go through cell division and we reduce the number of chromosomes. So we'll see how that works. But that allows us to start with a diploid cell and then produce haploid cells from it.
Yep.
All right. So if we look at the way this works for humans and all animals for sure, the plant life cycle, the fungus life cycle, those are sexually reproducing organisms. It's slightly different though. But all sexually reproducing organisms have part of their life cycle as diploid and part of the life cycle as haploid. For our life cycle, our diploid phase is much bigger. We would say it's the dominant phase. And the haploid phase is much smaller. Okay, so when we look at how this works, like I said earlier, your whole body is made of diploid cells that go through mitosis to grow and repair tissue and make new cells. And it is just the gametes that are haploid that would then combine to make a new diploid cell. So we do alternate between diploid cells and haploid cells, but the haploid part is a much smaller part of our whole life cycle there. So we do alternate between diploid and haploid. We just are much more sort of heavily weighted on the diploid part rather than the haploid part.
Like,
Still very important. What?
Like you're saying, like we have more diploid cells in our body than the haploid.
Way more, because of your trillions of cells, all of them are diploid except for maybe a few thousand egg cells, which are haploid. So way more diploid, yeah. And I'm talking about time too, like timing wise, there's just more time in that diploid part of life than the haploid part of life.
So like a haploid cell would be like our eggs?
That's it, yeah. Or the sperm from males, yeah. Those are the only two examples of haploid in us, yeah. Yeah. So again, we've learned about mitosis and how that cell division works. We have to talk about meiosis, like how we take diploid cells in our ovaries, in the male's testes. and go through cell division to produce those gametes that are haploid. So that reduction of chromosomes that we mentioned earlier, that's what we have to focus on. Meiosis is cell division, and again, like I said earlier, you're going to see a lot of the same stuff that we saw in mitosis, but there's going to be some distinct differences that we have to point out, and some of them I'm mentioning here. So when we look at meiosis, there's 2 rounds of division. We have what we call meiosis 1, and we go through prophase, metaphase, anaphase, telophase in meiosis 1, and then we have what we call meiosis 2. And so there's prophase 2, metaphase 2, anaphase 2, telophase 2. So 2 rounds of cell division. We did not see 2 rounds of cell division in mitosis. We just went through the whole thing and essentially made a copy of that cell in 1 round. But remember, we're trying to reduce the number of chromosomes in this. Another feature that's unique that we haven't seen and so that we haven't talked about before is synapsis, which is what this picture right here is trying to show. Synapsis is when the homologous chromosomes combine. They like fuse together. They join together. Do you remember what homologous chromosomes are?
2 chromosomes.
The same,
Yeah.
They're the same. Well, now, they don't come from the same place, right? Because where did they come from?
Mom and dad.
Yeah, you got 1 from mom, 1 from dad, but we say they're the same because they have the same genes on them, right? So it would be like, if you think back to when we talked about karyotypes, it'd be like chromosome 1 from dad, chromosome 1 from mom, right? They're homologous. Chromosome 2 from dad, chromosome 2 from mom, they're homologous. And then 3, 4, all the way through, right? So during meiosis, these homologous chromosomes find each other and they link up. They like stick together. That's what you see going on right here. So this might be chromosome 1 from dad and it's attached to chromosome 1 from mom. They have the same genes on them, and you can see these attachment points. They're showing how there's proteins that hold those 2 chromosomes tightly together. So the reason this becomes important is because now these things will move together as a unit. We call this structure a tetrad. Why do you think it's called a tetrad? Do you know what the prefix tetra means? It means 4.
Yes, let's go.
And we're looking at 4 sister chromatids. That's why it's called the tetrad. So you can see 1, 2, and then 3, 4 tetrads. Chromatids. That's why it's called a tetrad. But now this thing, once it's formed, will move as 1 unit. Did we see tetrads in mitosis? No. Did we see homologous chromosomes linking up with each other in mitosis? No, we didn't. I didn't mention that word tetrad before. I did not talk about synapsis before. We did learn about homologous chromosomes, but we did not see them coming together, right? So this is new to meiosis, and this is one of the big differences between meiosis and mitosis that leads to different results. Question?
So that's not a sister like thing?
Each of these, this is still 1 sister chromatid, another sister chromatid, 1 here, 1 here. So there's 4 sister chromatids, but the fact that they're all stuck together, that's unique to meiosis.
I thought we been seeing stuff like that too.
We saw 2 sister chromatids linked together. That forms 1 chromosome, but now we're seeing 2 chromosomes with their sister chromatids linked together.
So the blue is 1 sister chromosome, the red is another?
No, the blue's 1 homologous chromosome, the red's the other homologous chromosome. The blue has 2 sister chromatids, and the red has 2 sister chromatids.
So before we were only seeing 1 blue one or 1 red.
They weren't attached to each other. Yeah, they stayed separate. That's what I'm saying.
And now they're attached to a
And now they're together. Yeah.
And that's the synapse thing?
Well, synapsis is the process of them coming together, but the structure that forms is called a tetrad. Okay, so synapsis is the formation of this tetrad. It's when the 2 homologous chromosomes find each other and fuse, essentially. They bond together. Now, this is going to happen at the beginning of meiosis 1. So specifically, this synapsis occurs in prophase 1. And we'll look more specifically at prophase 1, so you'll see it come up again. But if you think back to mitosis, we didn't see this. We didn't see this in prophase or anything like that. Once the synapsis occurs and these tetrads form, it moves as 1 unit, and it does eventually get separated, but that looks different. Now, because these tetrads form, another thing happens in meiosis that we didn't see before, and it's called crossing over. Crossing over is the exchange of genetic material, like DNA, exchange of DNA, between these homologous chromosomes. So what I mean by that is like this chromosome and this chromosome are swapping pieces of each other. They're swapping DNA. And it's an even exchange. It works equally. But like you'll see by the end of crossing over, like this blue chromosome will have a little piece of red. And this red chromosome will have a little piece of blue. They literally swap chunks. They swap sequences. And again, that did not happen in mitosis. We didn't mention that before. But the fact that because these homologous chromosomes find each other and cling to each other, fuse together, they're so close to each other that it becomes easy for them to swap pieces. And again, that's what crossing over is, that exchange of the genetic material. And so that would also happen in prophase 1 when those tetrads form, when that synapsis occurs. So I'll mention that again as we go through these phases, but this is just a quick overview.
Crossing over only happens in the tetrad.
In the tetrad, yeah, between the homologous chromosomes, yes. Okay, and then the other thing that's important, I did mention earlier that we have these 2 rounds of cell division. Well, when we talked about mitosis, we saw how interphase occurs first, and then the cell divides, then it goes back into interphase, and the cell divides. Like, what we see in meiosis is that it goes through interphase before meiosis 1. And what did we say earlier is happening in interphase? You got G1, you got S, and G2. What's going on? You got growth, you got DNA replication, a little more growth, and preparing for division, right? So before meiosis 1, we'll go through interphase, the cell will grow. More importantly, it will copy its DNA, grow a little more. Goes through meiosis 1. But in between meiosis 1 and meiosis 2, there is no interphase. So there is no DNA replication between meiosis 1 and meiosis 2. And that, again, is one of the things that's going to help the cell reduce its number of chromosomes, among other things. So there's no interphase, there's no S phase specifically between meiosis 1 and meiosis 2. So that DNA does not get replicated there. And again, that's helpful because it allows us to take these diploid cells and reduce their number of chromosomes to haploid. We'll look at that in just a second. All right, so like I mentioned, when we look at meiosis and we break down the parts, you have 2 rounds of division. So you have meiosis 1, you have prophase 1, metaphase 1, anaphase 1, telophase 1. You have prophase 2, metaphase 2, anaphase 2, telophase 2. And we do have to walk through and see what each of those looks like, what's going on. But that won't be super difficult because we already know what mitosis looks like. We just have to point out the differences. Like I said, there is an interphase before meiosis 1, so we do have G1, S, G2. That DNA does get replicated before meiosis 1. But again, after meiosis 1, there's no interphase between telophase 1 and then prophase 2. There's no interphase there. So we're starting, wherever you see this term germline cells, that refers to a diploid cell that's going to become those haploid cells as a result of meiosis, just so you know. So those are the cells that we're starting with and that we're going to make egg cells, sperm cells with. All right, let's walk our way through the steps in meiosis. I don't have a slide specifically for interphase, but we know what happens in interphase. What did we already say happens in interphase? Cell grows, DNA gets replicated, preparing for division. That's what we need, right? So we're going to jump right into prophase 1 and see what's going on in prophase 1. All right? And so we have pictures that will help us to visualize this. But remember, some of the things will look similar to mitosis and some will look different, okay? So what is similar? Well, I see the spindle fibers forming. That happened in prophase of mitosis. I'm already familiar with that. The nucleus is going to disappear. That also happened in prophase of mitosis, so that we already know. But we do have to pay attention to what's going on with the chromosomes because they're going to behave a little differently here. Those chromosomes do form. So from interphase, we had chromatin. They do have to coil and become chromosomes. But what's different about prophase 1 is that those homologous chromosomes are going to find each other and form that tetrad. So you're going to have synapsis occurring. And you can see it, one tetrad right here, and there's another tetrad right here. These are not human cells. If these were human cells, how many tetrads would we see? How many tetrads? 23, right. Because we have two sets of 23, right? We have 46 chromosomes total. They would find their homologous pairs. That would give us 23 tetrads. So if this was a human cell, instead of 2 tetrads, I'd see 23 tetrads. But it's just easier to look at like this. So that synapsis occurs, those tetrads form. And because those homologous chromosomes are just stuck to each other, crossing over can also occur. That's where they exchange genetic material. And you can even see in this picture how this red chromosome has a little piece of blue and the blue chromosome has a little piece of red. So you can see where they exchanged that DNA and that crossing over occurs. So we definitely do need to know that prophase 1 is where those tetrads form, where that synapsis occurs. And because that tetrad forms, crossing over also happens here. So that's the two big things that are different between prophase 1 and prophase of mitosis. The fact that those homologous chromosomes pair up, form the tetrads, and then that allows crossing over to occur. The other stuff we see going on is the same. Spindle forming, nucleus disappearing, chromosomes condensing. We had seen all that already. That is not anything new. Okay? Good so far? Let's take a look at crossing over a little more specifically. The reason why it's important to talk about crossing over and we need to understand its usefulness, basically crossing over is one of the things that allows for genetic variation. And genetic variation is super important. Like when you look at a population, you talk about a human population, you talk about whatever plant population, whatever it is. Genetic variation will always lead to a more stable and a healthier population. So the more variety a population has, the more likely it is to survive different environmental circumstances, changes, whatever it might be. So variation is always good. Because crossing over occurs, it allows the chromosomes to get new combinations of genes. And again, you're increasing that genetic variation. That is always a good thing. Because what we're really doing is we're taking these two chromosomes that came from different people, right? If these are homologous chromosomes, one came from dad, one came from mom. But when they form a tetrad, they really form new combinations of genes on chromosomes that then get passed to the next generation. So we're creating all new varieties of genes, not necessarily new varieties of genes, but new combinations of genes that we got from our parents, and then we're passing those new combinations on to our kids when we make our gametes. So that's why crossing over is so important. It allows for new combinations in the chromosomes, increases genetic variation. That's always, always, always a good thing. So the reality is when you pass your chromosomes on to your kids, you're not passing a whole chromosome from your mom or a whole chromosome from your dad. You're passing on a chromosome that has a lot of genes from your mom, but then some from your dad too. Or a lot of genes from your dad, but then some from your mom. So it's not quite as cut and dry as we might think. We get new variations new combinations, I should say, of the DNA that we inherited that we then pass on to our kids. So that crossing over occurs in prophase 1. These tetrads will stay intact until we get through anaphase 1. So we'll see where they split up. But they will eventually get pulled away from each other and separated into different cells. So I mentioned crossing over. The site where that crossing over occurs, you can see right here, this site where you can see that exchange that swapping, is called the chiasmata. I'll write that down on the board. Chiasmata. It's CH actually. IASMMA. That's plural actually, I think, for chiasma, I want to say. But that's those sites where the crossing over actually occurs. So that actual exchange where they swap pieces of each other. Those are chiasmata. Very good?
Yes.
All right, so this was all stuff in prophase 1. Crossing over, tetrads, all the other stuff we're already familiar with prophase. Now let's look at metaphase 1. I would just look at this cell. How does this look different than metaphase of mitosis?
The two,
They're not
the
split
benefits,
apart.
oh, yeah.
Go ahead.
The two,
What do we see lining up? What do we call those? The tetrads, right? So in metaphase 1, we see tetrads lining up in the middle of the cell, not individual chromosomes, right? Because if this was metaphase of mitosis, this guy would be by himself, this guy would be by himself, these two would be separated too. So this definitely just looks different, but we still see things lining up in the middle of the cell, so that's the same. We still see chromosomes attached to spindle fibers moving chromosomes around, but this time it's not moving individual chromosomes, it's moving the tetrads, which are those homologous chromosomes that are linked together. So crossing over has already occurred. That happened in the last phase. The microtubules or the spindle fibers are attached to those homologs and pulling them so they line up in the center of the cell. So pay attention though, because we don't see the microtubules or the spindle fibers attached to each sister chromatid. We see it attached to the whole chromosome. And now we're moving the tetrads. So again, that's a difference. We're going to wind up splitting the tetrads, not splitting sister chromatids. So homologous chromosomes are lined along the metaphase plate, side by side. Again, that's your tetrads. And the order of those homologous chromosomes is going to vary. You're not going to have it set up to where all dad's chromosomes on the left, all mom's chromosomes on the right, and then they separate. It's going to be a mix of like some are lined one way, some are lined the other way. In fact, if we look at this picture, you can see all the different possibilities of how these chromosomes can line up. It's completely random, right? So we might have all blue on one side, all red on the other, or all red on one side, all blue on the other, or a combination, some blue, some red, some blue. So many different variations of how those chromosomes can line up. And the reason why we're mentioning this is because how these chromosomes line up in the middle is also going to determine how they get pulled away from each other. And so that means the way they line up is going to determine how those chromosomes get separated into those different cells, because these are going to go one way, these are going to go the other. So we can see how once these split, we're going to wind up seeing tons of different possibilities for the cells that form. The way these split, is this going to create identical cells after these chromosomes split? No, we are not going to see genetically identical cells. Meiosis does not make genetically identical cells. In fact, it is purposely designed so that we get more of that genetic variation. So this is another way that genetic variation is increased when we look at meiosis, is because just the way these chromosomes line up, you're going to wind up separating different chromosomes, like, again, different maternal, paternal in different ways, all kinds of random different orders that they could be separated. And so, again, just increases variation when we look at how meiosis works. Question, Harley?
So each tetrad thing, that's called a homologous, or like that's a different thing?
Homologous chromosomes link together to form the tetrad, or homologs link together to form the tetrad, if that's what you mean.
Okay, so whenever it says like whenever they're locked together, following crossover, that just means that they're like locking together.
We still have that tetrad, like, that's what we mean. We still see the tetrads, that's what I'm saying.
Okay.
Yeah. Okay? So, but now the tetrads are lined up in the middle. And so we know what's going to happen next. They're going to split, because that's what anaphase does. So let's take a look at anaphase. And now we can see how we have chromosomes being pulled away from each other to opposite sides of the cell. But again, it's not sister chromatids that are pulled away. It's the homologous chromosomes. So the homologous chromosomes that were linked together in a tetrad, Now that tetrad separates. You have one homolog moving one way, you have one homolog moving the other, right? So, how many chromosomes do we see now? We still see 4. If we looked at the tetrads, that would technically still be, like if I asked you right here, how many chromosomes you see, you still see 4. I didn't mean to confuse you by that, but keep in mind, these are still replicated chromosomes, right? What do I mean by replicated chromosomes? Why am I calling this a replicated chromosome?
Because it's replicated from your mom and dad.
Depends on what you mean by that.
I don't know what I mean.
It's like a variation of your mom and dad's.
No. Okay, so like these are replicated chromosomes, but these are not replicated chromosomes.
Because they're not two together.
That's what I mean. So these you don't see 2 sister chromatids together, right? But a replicated chromosome, you still see those 2 sister chromatids together, right?
So
Okay.
that's the pair if they're replicated.
You have to see 2 chromatids, yeah, together.
So, like, that doesn't break up the sister, that doesn't break up, like, the tetrad thing?
This did break up the tetrad. So before, you saw the tetrads lined up, right? Look at it. You see the tetrads lined up. Anaphase, now the tetrad's separated.
So now each one is, like, a different sister?
Well, they both still have 2 sister chromatids, right? You can see 2 here, 2 here, 2 here, 2 here, right? But we've taken those homologous chromosomes that were connected to each other, and we've separated them. Again, this is different than mitosis. Mitosis, we saw just a replicated chromosome separate its its chromatids, right? But now we're seeing a tetrad, and then the homologous chromosomes move away from each other.
So how is it still a tetrad?
It's not still a tetrad.
So what is it?
We would call these chromosomes, they're chromosomes. They're replicated chromosomes, but they're individual chromosomes.
So that's just, like, half of the tetrad?
It is half of the tetrad. Yeah. All right. So the tetrad splits, those chromosomes move away from each other, but again, they are replicated chromosomes. So we will eventually have to separate those chromatids, and we'll get to that in just a second. Now, when these chromosomes move away from each other, let's look at something. Let's start with this cell, the very first cell I showed you in prophase 1. How many chromosomes are in this cell? There's 4. Yeah. 1, 2, 3, 4. Can you see that?
Yes.
4 chromosomes. Question?
So the replicated chromosomes is just like a regular.
That just means it's a chromosome with 2 chromatids, yeah.
So just like a normal one?
Sure, that's what, yeah.
Okay.
Okay. So 4 chromosomes in this cell that we started with. This is a diploid cell. So if its diploid number is 4, what's its haploid number? 2. Diploid means 2 sets, so haploid means 1. So we're dividing. Diploid means 2 cells, 2 sets of chromosomes.
Yeah.
Think of like.
So what's its haploid? If our diploid number is 46, what's our haploid number? 23. So if this diploid number is 4, again, what's its haploid number? 2. Okay, so 4 chromosomes. When these cells split, how many chromosomes are going to be in each cell? 2. 2, right?
Oh.
Yes?
Yeah, that makes more sense.
So this is going to make haploid cells. Now, these are still, again, replicated chromosomes. They still have 2 sister chromatids, but it's still going to make haploid cells. We're okay with that?
Yes.
So what I'm saying is meiosis 1 makes haploid cells. I'm just pointing out why that is true. All right, anyway, homologs get separated. They move away from each other. And that's why we can say each pole is going to have just a haploid set of chromosomes. Because there's only 2 going this way, and there's only 2 going that way. Diploid was 4. Haploid's 2. All right, let's look at telophase 1 then. Much like telophase of mitosis, the nuclei are going to reform, spindle fibers are going to disappear, those chromosomes are going to uncoil. So we know all that. We learned all that from mitosis. What's different? Well, what's different is that these chromosomes are still replicated, meaning they still have 2 sister chromatids connected to each other. But now we're actually making cells. When these cells separate, like I said earlier, they're going to be haploid cells. Because now I only have 2 chromosomes here, I have 2 chromosomes here. Even though they're replicated, it's still just 2 chromosomes. So this is a haploid cell, and this is a haploid cell. They are already genetically different. Like the chromosomes here are not the same chromosomes here. So they're already genetically different. I have gone from a diploid cell to 2 haploid cells by the end of telophase 1.
Is it possible for them to end up the same on both sides?
It is, but not very likely. Okay. The way crossing over would have to occur, and then the movement to the opposite poles, like the odds of that happening are probably 1 in some kind of crazy, like 1 in a trillion type
Isn't it a
situation.
problem?
No, it wouldn't. All right, so after the end of telophase 1, remember we're not going to go through interphase again. There is no replication of the DNA. We move right into meiosis 2. So then if we look at meiosis 2, you go through again prophase 2, metaphase 2, anaphase 2, telophase 2. We already have haploid cells. We already reduced the number of chromosomes. What this is about now is taking those replicated chromosomes with the 2 chromatids and separating the chromatids into different cells. So the haploid has already been achieved. But we're going to see a lot of the same stuff that we've already seen. What's nice about meiosis 2 is that it really does look exactly like mitosis. So all that stuff that you saw, it looks exactly like mitosis. It's just mitosis of a haploid cell. Whereas all the other cells in our body, you do a mitosis of a diploid cell. This is mitosis of a haploid cell. So this one is going to look like there's nothing new or different. There's no crossing over. There's no tetrads. None of that. It looks just like mitosis that we had learned previously. So let's walk through it. Now remember, you're going through this with 2 cells because we formed 2 cells by the end of meiosis 1. So we're seeing meiosis 2 happen with 2 cells at the same time. And so we see spindle forming, the nucleus disappears, these chromosomes have formed, but again, they're haploid cells. But you can see the sister chromatids. We are not going to form tetrads. We're not going to have crossing over. We don't really have homologous chromosomes available in the same nucleus anymore because, again, these are haploid cells. There's only 1 set of chromosomes instead of 2. So there's no opportunity for homologous chromosomes to link up. But it looks just like prophase of mitosis. Spindle fibers attached to the chromosomes. The chromosomes will get pulled to line up in the middle of the cell. That's meiosis 2. I don't see tetrads. I see replicated chromosomes with their 2 sister chromatids lined up in the middle of the cell. And they're attached to the spindle fiber, and the spindle fibers are going to move them and then help to pull those chromatids apart from each other. When you look at anaphase 2, those chromatids have been pulled apart from each other. So now we would call those individual chromosomes. Remember that from mitosis? Once those chromatids aren't attached, as they separate, they are referred to as individual chromosomes. But now those are moving to opposite sides of the cell. So some are going to go down, some are going to go up. Now when these cells divide, how many chromosomes will they each have? When they divide, how many will each have? 2, right? And that's what we had said earlier as well. But the 2 that we had in each cell before was replicated chromosomes. Now they're not replicated anymore. They're like individual non-replicated chromosomes. So that's anaphase. The chromatids get pulled to opposite sides of the cell. And then telophase 2, we see nuclei reforming. Those chromosomes will uncoil. The spindle disappears. We will wind up with 4 separate cells by the end of telophase. They will definitely all be haploid. They will also all be genetically different from each other. So by the end of meiosis, unlike mitosis, you get 4 cells. The number of chromosomes has been reduced from diploid to haploid, and you've made 4 genetically different cells just because of the way those chromosomes lined up, the way crossing over occurred, the way the homologs and then the chromatids got separated into different cells. it creates that genetic variation. Question?
How did, like, can you go to the slide before? How did it split up into from the last, like, thing it only had two?
From here to here?
No, like the, I forgot what it's called, like the whole other, like meiosis one, or like meiosis one.
Okay, so you're talking about the cells or you're talking about the
The
chromosomes?
chromosomes, like you had two.
So at the end of meiosis 1, we have 2 chromosomes in each cell, right? Yeah. And then spindle will attach to each of those and pull the chromatids apart. Is that what you're asking? Does that make sense? So we'll separate the chromatids as we go through meiosis 2. So the chromosomes line up and then the chromatids get pulled away from each other.
After meiosis one, you have 23 in each cell. And
Yeah.
then you run it again, so then you have even less than that.
No, you wouldn't. The number doesn't get reduced in meiosis 2. It's just the fact that those 23 chromosomes you have by the end of meiosis 1, they still are replicated. They still have sister chromatids attached to each other. So meiosis 2 is about taking those chromatids and then separating them into different cells. Okay. So you've already done the reduction. Meiosis 1 is where the chromosome number gets reduced.
But
And
they're
then
still like sister.
but there's still sister chromatids attached to each other. We got to separate those in meiosis 2. Okay. So end result of meiosis. I think I just said this, but you get 4 genetically different haploid cells. That's what becomes your egg cells, or in males, their sperm cells. But the life cycles of other organisms like plants and fungi are slightly different. And so for them, they can actually take those gametes or those products of meiosis and then run mitosis with them. And they make a lot more cells. They can make many, many more gametes than what we would make. Or like in a fungi, those haploid cells form the basic structure of like the whole organism is haploid. So it's just a little different than the way our body works. Not super important, but in result, you get haploid cells that can either be used for reproduction or can be multiplied, so you have more of them, either way. Now, this process sometimes doesn't work properly, and I think we've mentioned this. I feel like we mentioned it kind of informally. There are some times where this process doesn't work properly, and it does usually cause some major problems. And so we refer to that, when we look at how meiosis can go wrong, we refer to that as non-disjunction. Non-disjunction is when these chromosomes don't get divided evenly into the gametes that are being made. So if our tetrads don't separate properly, or if the chromatids don't get separated properly, you're going to wind up having cells that have extra chromosomes or are missing a chromosome. And remember, these are the cells that form your gametes, your egg cells, your sperms, or male sperm cells. And so if those cells get used for fertilization, they don't have the right number of chromosomes. That's going to be a big problem.
Is it Down syndrome when you're missing one?
Exactly. Well, you have an extra, actually. Yeah. And so non-disjunction, the incorrect distribution of these chromosomes, leads to what we call aneuploid gametes. Aneuploid refers to the fact that they have the wrong number of chromosomes. So how many chromosomes should you have in your gametes, in your egg cells? 23, right? That's your haploid. You should have 23. If you were to have an extra, or if you were to be missing one, that's what we're referring to, the aneuploidy. Having an extra chromosome is referred to as trisomy. That makes sense, right? Mm-hmm. After fertilization occurs, I should say, because you should have 2 of everything, and trisomy means you would have 3 of something instead of 2. And the example of that is Down syndrome, which is what I have in this. This is a karyotype of an individual with Down syndrome. Down syndrome is trisomy 21. So this person has 3 of the 21st chromosome instead of 2. And you may think, well, having extra shouldn't be a big deal. Having extra of DNA or missing DNA is actually in both situations a big problem. So Down syndrome is actually one of the few examples of non-disjunction that doesn't lead to death. But there are severe side effects. So there's usually a heart defect, there's brain development problems, there's specific facial features that go along with it. So there are severe side effects. The severity of it can vary a little bit, but there are problems with it. Most of the time, if you have aneuploidy of almost any of these other chromosomes, especially the ones that are really big, because they have so much DNA on them, if you're missing one or you have an extra one, it's going to lead to a miscarriage. Like almost always, that's fatal and you can't survive it. So that would lead to a miscarriage, and statistically, they say that is very often why miscarriages occur. Not always, but that is very often why miscarriages occur. There is some type of chromosomal anomaly. You have an extra one, you're missing one. Most of the time that leads to death. Trisomy 21 is just one of the examples where it wouldn't lead to death, but there are severe side effects. And you can have trisomies and monosomies of the sex chromosomes too, and that wouldn't be fatal, but again, they would have side effects as well. So that stuff, because I do think we had talked about trisomy 21, is a result of the incorrect distribution of chromosomes during meiosis. That's why we're mentioning it here. It's when things go wrong in meiosis and those chromosomes don't get divided up evenly. All right, the last little bit we need to do is just talk about very clearly, because we've mentioned it along the way, but just to hit on what exactly is different between meiosis and mitosis that leads to these different results. Remember, mitosis, the whole point of that is to make an exact copy of a cell. So you copy DNA, you make an exact copy of that cell, and it has the same function. It works the same way. Meiosis has a different purpose. Meiosis is for taking a diploid cell and making haploid cells from it. So we have to reduce the number of chromosomes for that because we want to use those haploid cells to reproduce, to make our offspring. So there's 4 key events that lead to those different results when we compare meiosis and mitosis. First, the homologous pairing and the crossing over. So the synapsis, where those homologs connect with each other and form the tetrad, and then the crossing over during the with the chromosomes in that tetrad. We said all that happened in prophase 1, that we did not see in mitosis at all, but the formation of that tetrad and then the movement of the tetrad and the separation of the tetrad, that's what really reduces the number of chromosomes, and because of the crossing over leads to genetic variation that we see from meiosis. Second thing says the sister chromatids remain joined at their centromeres and they segregate together during anaphase 1. So the fact that, in meiosis 1, when the tetrad splits, you still have your sister chromatids together. They'll have to split later. But the meiosis 1, we're splitting the tetrad, not the individual chromatids. And that leads to a reduction in the number of chromosomes in the cells that we make. And again, that's what leads to our haploid cells being made in meiosis 1 instead of just, dividing and making diploid cells again. Third thing, kinetochores, the sister chromatids attached to the same pole in meiosis 1. Again, that's just referring to the fact that those sister chromatids stay attached. They move together to opposite sides. They haven't been separated yet by the end of meiosis 1. That happens in meiosis 2. Different than mitosis because obviously we saw those tetrads separate and get pulled away from each other in mitosis. Fourth thing, DNA replication is suppressed between meiosis 1 and meiosis 2. We said there really isn't an interphase between meiosis 1 and meiosis 2. So that DNA does not get replicated, doesn't get copied. We're trying to reduce the number of chromosomes here. We don't want to copy them again. So that DNA replication happens before meiosis 1 and not before meiosis 2. Just a comparison, I know we already saw this, but look at how in meiosis 1, metaphase 1, your tetrads line up in the middle of the cell, then the homologs are separated. Metaphase of mitosis, each individual chromosome lines up by itself, and the sister chromatids are separated. So again, this was what allows for that reduction in number of chromosomes by the end of meiosis 1. This is what allows us to go from diploid to haploid by the end of meiosis 1. And we've already walked through these pictures, but this is the nice diagram, nice big diagram that walks you through what meiosis looks like versus what mitosis looks like. Remember we said meiosis 1 is the reduction division. We take a diploid cell, and by the end we produce 2 haploid cells. So you can see this is a cell with 4 chromosomes. By the end of that, I've got 2 cells with 2 chromosomes. But when I look at mitosis, by the end of mitosis, I've got 2 cells and they both still have 4 chromosomes. So I've reduced the number of chromosomes here because of the way those tetrads formed and then the homologs got pulled apart. Meiosis 2 was the one that we said looks exactly like mitosis, but it's mitosis of a haploid cell. So these cells are already haploid, and they go about dividing those sister chromatids. You wind up with 4 haploid cells that are each genetically different. And that would be used to make our egg cells. Question? All right, we did it. We did the whole chapter.
Oh my gosh, it's Taylor time.